It looks like a chain of crystals. Thing right. More crystals in the shape of hexagons. These come from different people, issuing from the lesions, many embedded in fibers or tubes. They are clearly geometric, glittery, very like piezo reactors. Here's a three-dimensional hexagon found with a tissue filling like a sandwich. You can see the fiber below it with a small hexagon inside. Let's look at one of the previous hexagons, and I'm going to go back to that slide because we didn't really look at that one for very long. The one that I'm going to show you again is up at the left, top left, the blue one with the black. That's actually a very advanced little machine. And then the two below it are hexagons embedded in this biofilm that hardens. The biofilm starts as a glue and then it becomes very hard. And you can see that there are these very different shapes. There's a pyramid shape, and then the front and back can be entirely different. And just remember that crystals are one of the best piezoelectricity generators. So I'll go to this one. Okay. So this was the little one you saw in the top corner before. It's a composite of several crystals hinged with bioglue that glow and light up when touched with a needle. This is what Jan Smith calls a nanomachine, a device that has to have specific microelectronic functions and capabilities. I'm going to show you a video where you can watch its behavior. It's the size of a single letter. You can play the video. It's the size of one letter on a US penny, this thing. So it's smaller than a sesame seed. And as she opens it, that's a sewing needle, opening it up. You can see that the crystal inside is clear and that it's on some kind of hinge, which she told me was actually bioglue hinge. She'll move it. And you see this mirrory interior. So it springs like that, and then the inside of it has this very bright, she said it was like aurora borealis colors that changed with movement. And this was the same device that had a glyph on the back. And you can see on the left how it's layered when it's sandwiched together. Okay, here are plaques. These are hard silica-like forms that shatter under pressure. And remember, frequency is pressure. So just touching with a needle caused this blue plaque to break into smaller fibrous replicas of itself, and it created a colony. That's the fourth picture. So that precursor was number one, and then it was that number two shows a more fully formed version, and then it shatters and it appears to make smaller replicas. She's broken a lot of them. She gets a lot of them, and this is what she's observed. And from everywhere come the fibers. The fibers are filaments. They're hollow tubes or tubules. The goo is a kind of biofilm that eventually hardens into these fibers. And in the fiber masses are the crystals and the nanotechnological devices, grooved, marked with colored dots, at times etched and labeled. And I talk about these at length in the other talk, which you can get at my table, or you can see it on YouTube, the talk I did at ConspiracyCon. So now, here is a very important biology lesson. Tubes are the fundamental unit of organic design. Tubes are fundamental to natural forms. The human body is a tube. It's a skin stuffed with bones and organs. Plants are tubular constructions, from the trunk of a tree to its branches, twigs, and leaves. The leaves consist of a network of fibers in and along which the business of life is performed. So organic life depends on tubular networks and is created in and from tubes. Mark Krasnow is a biochemist and medical doctor at Stanford. He has mapped the 5,051 airways of a mouse lung, all the sprouting and branching that expands a simple embryonic tube into a mass of filament fine airways that pour oxygen deep into the animal's body. This, he says, is the beginning of understanding organogenesis in mammals, how the growth or branching of tubules leads to organ formation. The lungs, for instance, begin as two small buds, almost like a wishbone, that branch into more buds, growing and branching over and over into a system of airways in a set of adult organs. The tube is an embryonic form. Now look at a picture of Morgellons tissue that was kept under a slide for 3 months. You can see the fiber network that grew in the slide all by itself from a piece of tissue. It didn't look like this in the beginning. Fibers are an integral Morgellons form, just as they are integral to biological life. And fibers, tubules, are hollow, meant to have some function to contain and produce something. Here's a picture of a fiber at 5600 magnification that was cultured by Clifford Carnicom with a visible internal structure mass that's on the left and a branching bud that's on the right. Here's another Clifford Carnicom discovery that is guaranteed to cause concern. Filaments that contain red blood cells and also are observed to be producing red blood cells. And here's what sends this through the roof. These filaments are not just found in people with Morgellons, they're found in everyone, and they can be cultured to produce the same things, red blood, for instance. So you have to ask yourself, what's this doing in our bodies? This is all engineered stuff, filaments that have no match to nature, that are engineering blood. And we all contain these engineered materials. Now I'm going to throw in a video I found on carbon nanoforms. In it you'll see similarities to the Morgellons tubules, their buds and their insides. And you'll also see the hexagonal skin on what are called fullerenes, named after Buckminster Fuller of the geodesic dome.